He proposed their existence in 1865. At that time, no cellular structures were known that could house for these imaginary units. Today, we can show that genes, mental structures are located along chromosomes, and we can see the locations of a particular gene by tagging chromosomes with a fluorescent dye that highlights that gene. Chromosomes, like structures that appear in nucleus during cell division. In an interphase, cell chromosome become uncondensed and look like very fine network called chromatin network. Chromosomes were first observed by German embryologist Walter Fleming in 1882 when he was examining the rapidly dividing cells of salamander larvae. The term chromosome was proposed by Wilder, which literally means colored bodies. Since their discovery, chromosomes have been found in the cells of the eukaryotes. However, in prokaryotic cells, a single DNA molecule is also referred as chromosome. Number of chromosome. The number of chromosome varies from species to species, and usually it is characteristic feature of many species. Penicillium, a fungus, has only one pair of chromosomes, while some ferns have more than 500 pairs. Many species have two sets of chromosomes in their somatic cells, hence called diploid, while some may have more than two sets of chromosomes. They are called polyploid, tetraploid, hexaploid. The term diploid is referred to the number of chromosomes exactly half than the somatic number of chromosomes. Gametes and spores are usually haploid cells. A haploid cell may be monoploid, one set, diploid, two sets, triploid, three sets, and etc. Number of chromosomes in human and weight. Human 46 2n number, 23 1n. Monoploid 23 weight 42 6n number, 21 3n number, 7. Structure of chromosome. A typical chromosome consists of strands called chromatids. Each is made up of long DNA molecule which is highly coiled along with histone proteins. Generally, both chromatids are attached with each other at a point known as centromere or primary construction. So each chromosome shows two arms regions from centromere to an end. Some chromosomes may have another point of union along the length of chromatids called secondary construction or nuclear organizer. It gives rise to nucleoli during interphase. At least one pair of homologous chromosomes possess nuclear organizer region. Besides secondary construction, the end becomes a nub-like structure called satellite. This region has useless sequence of DNA called junk DNA. The terminal ends of chromosomes are called telomeres, which prevent the two chromosomes to attach with each other from their ends. On the basis of position, centromere along the length, a chromosome may be called metacentric, centromere located in the center, submetacentric, centromere located slightly away from the center, acrocentric, centromere located near the end, telocentric, centromere located at an end. Composition and organization of chromosome. Generally, a chromosome is made up of 40% DNA and 60% protein. In the cell, which is ready to divide, the chromosome has two identical DNA molecules. For example, each chromatid has one DNA molecule. An average-sized human chromosome has approximately 5 centimeter long DNA, which consists of about 140 million nucleotides. So can you imagine how such a huge molecule fit into a tiny chromosome? DNA is negatively charged molecule because of phosphate groups. Therefore, it has strong affinity to histone proteins, which are positively charged, unlike many other proteins, due to the abundance of some basic amino acids such as arginine and lysine. There are five types of histone proteins: H1, H2A, H2B, H3, and H4, found in the chromosome. During S phase of cell cycle, DNA and histones are completely disorganized from each other, but after DNA is replicated, both DNA and histones begin to organize again. The process of condensation remains continuous till the cell undergoes division and the chromosomes are appeared. The organization of chromosomes occurs in four levels. Just after the completion of DNA replication, each DNA molecule of enormous length begins to coil around a stone core. In this level of organization, about every 200 nucleotides of the duplex DNA wrap twice around the core of 8 histones, two of each H2A, H2B, H3, and H4, thus forming a complex known as nucleosome. While H1 is associated with a small segment of DNA, linker DNA, between every two nucleosomes. In this way, the whole DNA, 2 nanometer thick, is turned to a chain of beads-like appearance called nucleosome string, 10 nanometer thick. Immediately, the nucleosome string begins to coil again about its axis to form yet another thick fiber of 30 nanometer called chromatin fiber or solenoid. During G1 and G2 phases, chromosomes are found in this level of organization. The chromatin fiber shows two regions, heterochromatin and euchromatin. Heterochromatin is highly condensed and unexpressed region, while chromatin is non-condensed, and the genes of this region are also expressed. When cell undergoes division, chromatin is also condensed so that a uniform chromatin fiber is established. When cell division begins, the higher order coiling of chromatin fiber gives rise supercoil which has diameter of 200 nanometer. Immediately, supercoil established into the chromatid of 700 nanometer as a result of further coiling around itself. Concept of gene historical background. Charles Darwin first conceived the idea of hereditary units when he published his theory of pangenesis in 1868. In this model, circulating units called gamules are accumulated in gonads and transmitted to the offspring. This theory was discredited by experiments performed by Francis Galton in the 1870s century. Galton used blood transfusion in rabbits to show that the alleged gambules in one rabbit blood did not alter the heredity of the recipient rabbit's blood. In the 1890 century, Hugo de Oris took the term pangenesis and framed it to pangene for the assumed units of inheritance. He argued that pangenes remained inside the cell and did not migrate. It was this theory of intracellular pangenesis that led Dioris to independently find what Gregor Mendel had discovered 30 years earlier in his work. With contrasting traits in garden peas, there are units of inheritance that are transmitted by reproduction. Wilhelm Johnson introduced the term gene to replace several contending and misleading terms for the basic unit of heredity. In 1909, the term genetics came earlier, when William Batson coined the word in 1906 to represent the new field that studied heredity and variation and evolution. Modern concept of gene. It is believed that the modern concept of gene was given by Mendel while his experimentation on pea plant. According to Mendel, each trait in the pea plant is controlled by discrete units, what he referred as Elementals are factors. In the modern terminology, Mendel's factors are called genes. Since the rediscovery of Mendelian work in 1900, the nature of and expression of genes is being explored by various geneticists all over the world, and this job is still continued. But now we know that a gene is composed of nucleotide sequence of a short segment of DNA, which encodes the sequence of amino acid of a particular polypeptide. Where do genes reside? These genes are found in the chromosome. The position on a chromosome where a gene is located is often referred to as a locus. Most genes exist in alternate versions or alleles, with differences at one or more nucleotide positions in the DNA. The alleles related to the same trait are occupied on the same locus. For example, gene of ABO blood group I exist in three different alleles, IA, IB, small I, that are found on the same locus at chromosome number 9. Structure of a gene. We have already discussed that a gene is a particular segment of DNA in which nucleotide sequence determines the sequence of amino acid of a polypeptide. A gene usually has regulatory regions and a structural region. The regulatory region located to the 5' of coding strand of the gene is called promoter that controls the binding RNA polymerase during transcription. Other regulatory region, the terminator is located to the 3' upcoding strand of the gene. The terminator region causes the RNA polymerase to stop transcription. The region between promoter and terminator is structural region of the gene that comprises information genetic code for a particular polypeptide or functional RNA. In eukaryotic gene, the information of structural region is interrupted by non-functional sequence called introns, where the functional sequences are called exons. There is no such pattern of introns and exons in the prokaryotic DNA. In prokaryotes, many adjacent structural genes that synthesize different polypeptides are regulated by same promoter and terminator regions. Such a group of genes is called a peron. Chromosome theory of inheritance. The chromosome theory of inheritance is the idea that genes, units of inheritance, which are physical in nature, are found in the chromosome, so chromosomes act as carriers of heredity. First time this idea was put forward in 1900 by German geneticist Karl Kearns in one of the paper announcing the rediscovery of Mendel's work. But he had no supportive evidences for this idea. Therefore, actual credit of this theory goes to both Walter Sutton, an American who at that time was graduate student, and Theodor Beury, a German biologist in 1902. These scientists recognized independently that the behavior of Mendel factor gene is parallel to the behavior of chromosome at meiosis, which is pointed out in the following table. In addition to the above evidences based on parallel behavior between genes and chromosomes during meiosis, we can also analyze the mechanism of sexual reproduction, which involves the initial union of two cells, egg and sperm. If Mendel's model is correct, then these two gametes must make equal hereditary contribution. Sperm, however, contain little cytoplasm during fertilization and only contribute nucleus to the zygote. Therefore, the hereditary units must reside within the nucleus of the gametes, whereas chromosomes are also found in the nucleus. Besides above mentioned parallel behavior between genes and chromosomes during meiosis, this observation also indicates that gene would be present in chromosomes. Many investigators of that time pointed out that a serious objection on certain theory. According to that, let we accept that Mendelian traits are determined by the factor genes located on chromosomes. And if the genes are segregated due to segregation of chromosomes and a dependent assortment of genes as reflected by the independent assortment of chromosomes and meiosis, why is it that number of genes that assort independently of one another and a given kind of organism is often much greater than the number of chromosome pairs that the organism possesses? But later on, this objection was cleared after the discovery of linkage by historical experimentation of T. H. Morgan in 1910 and Drosophila. Behavior of chromosome, behavior of genes. Diploid cells before meiosis have two copies of each chromosome, homologous pairs, while gametes after meiosis have only one. For example, in pea plant, diploid cells have seven pairs of homologous chromosomes, while gametes have single seven chromosomes. Behavior of gene. According to the Mendel, diploid cells have two copies of each gene, pair of allele, while gametes have only one. For example, in pea plant, diploid cells have pair of alleles for gene like RR, and TT, while gametes have single R, capital R or small r, Y or small y, capital T, small t. Homologous pairs of chromosomes segregate during meiosis. According to Mendel, pair of genes for each trait also segregate from each other during meiosis. For example, RR. During meiosis, each pair of homologous chromosome orient on the metaphase plate independently of any other pair so that at anaphase each pair assert independently of the other. According to the Mendel, alleles of one gene pair also assert independently to the allele of other gene pair during meiosis. For example, capital R, small r, capital Y, small y. Genotype as a result of independent assortment can form four types of gamete. For example, capital R, capital Y, small r, capital Y, and small r, small y. Marcus' work with respect to the gene linkage and inheritance of eye color and Drosophila mangostar you have already been studied in the previous chapter. DNA as hereditary material. When the chromosome theory of inheritance was confirmed in 1910, geneticists started think over the issue that in which form the hereditary units are found in the chromosome. It was known that chromosome contain both DNA and protein. On which of these was the hereditary information written? Over a period of 30 years, starting in the late 19th century, a series of investigators addressed this issue, resolving it clearly. Here we are going to learn three experiments, each of which yield a clear answer in a simple and elegant manner. Griff's experiment. These experiments are very important for DNA. In 1927, British microbiologist Frederick Griff made a series of unexpected observations while experimenting with Streptococcus pneumoniae, which are found in two types. One of its type has a polysaccharide capsule. Its colony appear as smooth or shiny, and hence it is called S-type. The other type forms a rough colony due to the absence of polysaccharide capsule. This is referred as R-type. When Griff injected healthy mice with a strain of S-type, all the mice did. But when he injected similar mice with a strain of R-type, the mice showed no ill effect. On the basis of these observations, Griff made a hypothesis that virulent effect of S-type might be associated with polysaccharide capsule as the R-type that lack a capsule appeared non-virulent. Chromosomes and DNA. As spinal control, he blended living R-type with heat-killed S-type. Both of these strains were already confirmed as non-virulent. When he injected this mixture into the healthy mice, unexpectedly, the injected mice developed disease symptoms and many of them died. The blood of the dead mice was found to contain large number of living S-type virulent bacteria. On the basis of these unexpected observations, Greth concluded that somehow the information specifying the polysaccharides, capsule and virulence had passed from the heat-killed S-type bacteria to the live R-type. Once in the control mixture transforming them into live S-type virulent bacteria that killed the mice. This transfer of genetic material from organism to another by which genetic makeup of recipient is altered is called transformation. Average experiment. The agent responsible for transforming R type to S type went undiscovered. Until 1944, in a classic series of experiments, Oswald Avery, along the Golan, MacLeod, and MacLean McCarthy characterized what they refer to as the transforming principle. They first prepared mixture of dead S-type and live R-type Streptococcus pneumoniae that Greth had used in his last experiment. Then they removed as much of the protein as they could from the preparation by treating it with protease enzyme, eventually achieving 99.98% purity. Despite removal of nearly all protein, the transforming activity was not reduced. In the second attempt, all the RNA contents were removed with the help of RNase enzyme. But transformation remained continued. Finally, when mixture was treated with DNase enzyme in order to remove DNA contents of S-type, the mice injected with such a mixture in which DNA contents of S-type had removed developed no ill effect because no transformation occurred. In this way, it was confirmed that the transforming agent in the great experiment was DNA. Hershey-Chase experiment. Hershey and Chase experiments. Soon after the average results, another very convincing experiment on bacteriophages were performed by Alfred Hershey and Martha Chase in 1952, which was difficult to ignore. Bacteriophages are viruses that attack upon bacteria. Their body consists of DNA and protein. During infection, they multiply in the host and their many copies emerged within 20 to 25 minutes. It was not known till 1952 that either DNA or protein which possesses hereditary information of bacteriophages. Even scientists were not sure that during infection the whole viral particle entered the host body or only its DNA protein get entry. In 1952, Hershey and Chase set out an experiment to identify the viral part of that as injected into the host body at the start of infection. For this purpose, they labeled the DNA of bacteriophages with a radioactive isotope of phosphorus P and also labeled their protein codes with radioactive isotope of sulfur. The labeled viruses were permitted to infect bacteria. So, after the infection, bacterial cells were separated from media contents with the help of centrifugation technique. Then media contents and bacterial cells were analyzed for the activity of P and S. In this analysis, P32 was found in the bacterial cells while S was found in the medium. These observations clearly showed that during infection, P-labeled DNA of bacteriophage was injected into the bacterial cell while its S35 labeled protein code remained outside subsequently. Many viral particles released outside the host based on these observations. Hershey and Chase claimed that the virus DNA, not the virus protein, was responsible for directing the production of new viruses. DNA replication. The process of self synthesis of DNA molecule is called DNA replication. This process occurs only once in S phase during the life cycle of a cell. The molecule of DNA which is replicated is called parent DNA, while the molecule produced in the process are called daughter DNA. A parent DNA molecule after replication gives rise to daughter DNA molecule. How duplex DNA can replicate? Scientists started struggle to find the answer of this by the discovery of DNA structure. Overall, three different models to explain the replication process were come forward. Semi-conservation model. This model was given by Watson and Crick, who also proposed that the structure of DNA. According to this model, the parent DNA molecule becomes unwound and lost its base pairs unzipping. Both strands act as templates that allow the formation of new strands. As a result, two daughter DNA molecules are formed with one old and one new strand, hybrid DNA. In this way, the parental strands are partially conserved in what daughter DNA molecules. Conservative model. According to this model, the parental DNA remains intact in its duplex state, while a daughter DNA molecule with both new strands is established. In this way, the parental DNA is fully conserved in the next generation. Dispersive model. This model predicted that the parental DNA would become completely dispersed into fragments, which will be mixed with new nucleotide fragments, and this way the daughter DNA molecule would be mixture of old and new fragments. Michelson-Stahl experiment. The three models of DNA replication were evaluated by Matthew Michelson and Franklin Stahl of the California Institute of Technology in 1958. In this experiment, it was concluded finally that the replication of DNA occurs according to semi-conservative model. Culturing of bacteria. They grew bacteria in a medium containing heavy isotope of nitrogen, N, which became incorporated into the basis of bacterial DNA. After several generations, the bacteria were shifted to three separate plates which were already contained N14 medium. Sampling of DNA. Three DNA samples were taken from bacteria shifted from N medium, N15 medium to N14 medium. First samples was obtained from first plate just after the transfer of culture called sample at 0 minutes. The second sample was taken from second plate after 20 minutes, called sample at 20 minutes. And third sample was taken from third plate after another. Three DNA samples were taken. Three DNA samples were taken from bacteria shifted from N15 medium to the N14 medium. First sample was obtained from first plate just after the transfer of culture. Card sample at 0 minutes. The second sample was taken from second plate after 20 minutes. Card sample at 20 minutes. And third sample was taken from third plate after another 20 minutes. Card sample at 40 minutes. In addition to these, a control sample was also taken from the bacteria which were grown separately in N14 medium. Centrifugation of DNA samples. The DNA samples were dissolved in cesium chloride (CaCl) solution and then spun at a very high speed in an ultra centrifuge for many hours. The cesium and chloride ions tend to be pushed by centrifugal force towards the bottom of the tube. Results of centrifugation. DNA control sample was appeared latest as formed sediment at the top of test tube, while DNA sample at 0 minute was appeared heaviest as it formed sediment at the bottom of test tube. The DNA of sample at 20 minute formed sediment intermediate level to that of control sample and sample at 0 minute, where a sample at 40 minute had two sediments, one at the top and other at an intermediate level. Interpretations of results. Michelson-Stahl interpreted the result as follows. The DNA of control sample appeared lightest because it had both strands of N14, whereas DNA of sample at 0 minute appeared heaviest because it had both strands of N15. But after the first round of replication, each daughter duplex was a hybrid possessing a strand of N14, one of N15, so it formed sediment at intermediate level. When this hybrid duplex replicated in second round of replication, it contributed N15 strand to form another hybrid duplex, N14 strand to form a light duplex containing both N14 strands. That is why this sample formed two sediments. On the basis of above mentioned result, they claimed that the DNA replication is semi-conservative. Semi-conservative process of DNA replication. Although DNA replication is continuous process, but here we are going to discuss it as different phases for our convenience. Initiation phase. The initiation phase is characterized by the formation of replication bubbles and replication fork, which are formed at a particular site called origin of replication site. It is a specific sequence of nucleotides along the length of DNA from where process of replication begins. In eukaryotic DNA, there may be more than one origin of replication sites, but in prokaryotic DNA, there is only one origin of replication. Replication bubble is formed when DNA gyrase, topoisomerase, and DNA helicase enzymes work at origin of replication. DNA gyrase opens the turns of DNA duplex, so the DNA is converted from spiral ladder-like form to straight ladder-like form. At the same time, DNA helicase breaks down the base pairs of DNA, so the two strands gradually separate from each other and give a bubble-like appearance at origin of replication. After the breakdown of base pair, the single strand of DNA are prevented to pair up again by specific proteins called single-stranded binding proteins (SSB). Both single strands of DNA will act as template strands in the next phase and direct the synthesis of data strands along themselves. Each side of replication bubble is now termed as replication fork, extension polymerization phase. Extension or polymerization is referred to the formation of data strand leading or lagging strand along the template strand. The data strand are actually synthesized by DNA polymerase enzyme, but this enzyme cannot work unless some nucleotides are arranged on template. For this purpose, primase enzyme is involved to arrange some nucleotides on template strands. Such short fragments of few nucleotides are called primers. Each primer is short oligonucleotide strand of RNA act as start site for activity of DNA polymerase. After establishment of primer, synthesis of daughter strand begins by DNA polymerase enzyme. There are three different forms of this enzyme. DNA polymerase 1. It performs an important role in termination phase of replication, so it provides a support to DNA polymerase 3 in the main replication process. DNA polymerase 2. It involves in the repairing process of DNA damages during the lifetime of a cell. DNA polymerase 3. It is the main enzyme that synthesizes both daughter strands along the template during replication process. Mechanism of DNA polymerase 3 activity. This enzyme is a huge dimer molecule. It consists of two units that further consist of several subunits. DNA polymerase 3 cannot initiate replication process. It can add a nucleotide onto only a pre-existing 3OH group and therefore needs a primer to perform its polymerase activity. It's always add nucleotide at 3 end. A primer, so the direction of replication becomes 5 to 3 end. One of its subunit also possesses ability to remove wrong nucleotide if it is added mistakenly. This ability is called pro-upgrading. Base units of DNA polymerase 3 are interlinked by a small polypeptide chain. First unit work on one template and continuously synthesize a data strand toward replication fork. This continuously growing data strand is called leading strand, while the second unit work on other template and synthesize another data strand away from replication fork. As the two units are interlinked, so the second unit is allowed to polymerize data strand up to a specific length, then it has to jump back 100 to 200 nucleotides in the prokaryotes and 1000 to 2000 nucleotides in eukaryotes to a new primer to perform polymerization again. Therefore, this data strand grows discontinuously away from the replication fork by forming short fragments interrupted by primers called Okazaki fragments. This continuously growing strand is called lagging strand. Termination phase. Termination phase is characterized by the replacement of primers by DNA nucleotide and joining of Okazaki fragments and lagging strand to form a continuous strand. The replacement of primers by DNA nucleotide is carried out by DNA polymerase 1 that has dual function. Beside polymerase, it has act as exonuclease attached to the three-end Okazaki fragment where it adds DNA nucleotide so that it can extend, while on the other hand, it cleaves nucleotide from five-end of primer. And this way, primers are removed and each Okazaki fragment is extended up to the next Okazaki fragment, but they do not join together. The joining of Okazaki fragment is carried out by the DNA ligase enzyme that finally constructs phosphodiester bond between Okazaki fragments so a continuous strand is formed. Gene expression. Central dogma of gene expression. All organisms use same basic mechanism of reading and expressing genes, which is often referred to as central dogma. And this mechanism, the genetic information that resides in DNA, first flows down into the messenger RNA by process of transcription and then convert into protein by process of translation. Transcription. This is the process in which a messenger RNA copy of DNA sequence encoding the gene is produced with the help of an enzyme. RNA polymerase process of transcription is completed in three phases: initiation, elongation, and termination. Initiation phase. Transcription begins with the binding of RNA polymerase at promoter region. Promoter is a regulatory region of the gene which provides binding sites for RNA polymerase. It is located towards the 5' of coding strand in prokaryotes. There are two binding sites are located in promoter TA, TAAT, also called 10 sequence, and TTGACA, also called 25 sequence, whereas in eukaryotes, TATA box, Tata box, also called 25 sequence, and GAAT box, also called 70 sequence. Names of these sequence minus 10, minus 35, minus 25, minus 70 refers to position that these sequences are located. Therefore, the initiation site of structural region of the gene. RNA polymerase consists of four subunits: beta, beta, and sigma. Only the first three subunits are required for polymerase activity and are considered the core enzyme, while the sigma factor is required for RNA polymerase. To bind to the promoter, it is similar to DNA polymerase in that it also adds nucleotides to the three end of the growing polypeptide chain, but unlike DNA polymerase, it does not require primer to perform polymerase activity. In prokaryotes, only one type of RNA polymerase is found, while in eukaryotes, there are three types of RNA, namely RNA polymerase 1, which synthesizes rRNA, RNA polymerase 2, which synthesizes messenger RNA, RNA polymerase 3, which synthesizes transfer RNA. As the RNA polymerase binds to the promoter, DNA duplex become unwind. Base pairs are broken down and a bubble-like structure, the transcription bubble, is appeared. Elongation phase. As the RNA polymerase binds to promoter, sigma factor is released and remaining core enzymes extend the polymerization of ribonucleoside to initiate polymerization. One of the two strands of the gene act as template for transcription. This template strand is also called antisense because the messenger RNA is complementary to the strand. The outer strand of the gene is called coding or sense strand. An elongation phase, RNA polymerase keep on moving from 5 to 3 direction toward the terminator region. Beside it, transcription bubbles also moves along the DNA, leaving the growing RNA strand protruding from the bubble. This event continues till the RNA polymerase reaches the terminator region of the gene. Termination phase. The sequence of terminator region of the gene stops the synthesis of messenger RNA. The terminator region consists of a series of GC base pairs followed by a series of AT base pairs. The portion of messenger RNA which is transcribed in this region project to form a loop-like structure called GC hyphen followed by a small tail of AU nucleotide. The GC hairpin causes the RNA polymerase to stop the synthesis of RNA. Post-transcription modifications of messenger RNA. Post-transcription modification is a process by which primary messenger RNA transcript is converted into mature RNA or functional RNA. This process is only associated with eukaryotic transcription because in prokaryotes the messenger RNA that is synthesized during transcription is ready for translation into protein due to the absence of definite nucleus. On the other hand, eukaryotic messenger RNA is produced inside the nucleus, is not immediately ready for translation because it has to pass comparatively long distance to reach ribosomes in the cytoplasm. During this journey, some enzymes like phosphatases and nucleases can degrade messenger RNA before its translation. Another problem is that a newly formed eukaryotic messenger RNA comprises many non-protein coding sequences. As described earlier in this chapter, that eukaryotic genes have a pattern of exons protein coding sequence and introns non-protein coding sequence. So these non-protein coding sequences from a primary messenger RNA transcript are to be removed. Post-transcription modification is therefore involved to events. Addition of cap and tail to protect it from degradation and RNA splicing to remove non-protein coding sequences. Addition of cap and tail. A cap and tail are added to both ends of primary messenger RNA so that the molecule may remain stable and cannot be degraded by phosphatases and nucleases during long journey to ribosome. A cap is in the form of 7-methyl GTP, which has lagged from its 5 to the 5 end of messenger RNA modification also takes place at the opposite end of the RNA transcript in the form of small chain 30 to 500 adenine nucleotides called poly A tail, which is attached to the 3 end of messenger RNA. These two modifications prevent the messenger RNA to be degraded by phosphatases and nucleases. RNA splicing. A newly emerged eukaryotic messenger RNA is very long. It contains both exons and intron sequences. Introns are non-protein coding sequences which are to be removed from primary messenger RNA before its translation on ribosome. This removal of introns and maturation of primary messenger RNA to secondary functional messenger RNA is called RNA splicing. Genetic code. The sequence of nucleotides and DNA or RNA that determines the specific amino acid sequences of the protein is called genetic code. It is the biochemical basis of heredity and nearly universal in all organisms. The genetic code is stored on one of the two strands. The coding or sense strand of DNA molecules is a linear, non-overlapping sequence of the nucleotide. The genetic code is a coded language which is based on an alphabet consisting of only four types of nucleotide: adenine, guanine, cytosine, and thymine, which are variously arranged to form code words called codons. Usually, a codon specifies an amino acid, so the order in which codons are arranged in messenger RNA determines the order in which amino acids are arranged in a protein. Each code word codon is a unique combination of three letters that will eventually be interpreted as a single amino acid and a polypeptide chain. There are 64 code words possible from alphabet of four letters. One of these code words, the start of codon AUG methionine, begins all sequences that code for amino acid chains. Three of these codes words act as stop codons UGA, UAG, UAA that indicate that the message is over. Since these three codons do not encode any amino acid, hence called nonsense codon, while all the other sequences that encode specific amino acid are called sense codons. These are amino acids: phenylalanine, leucine, isoleucine, methionine, valine, serine, proline, threonine, alanine, tyrosine, histidine, glutamine, asparagine, lysine, aspartic acid, glutamic acid, cytosine, streptocodan, tryptophan, arginine, serine, arginine, glycine. Chromosomes and DNA. Some amino acids are only coded for a single codon, while some others are coded for by up to 4 codons, and amino acid leucine and serine are encoded by 6 codons. Degeneracy of the genetic code is an important characteristic which refers that an amino acid can be encoded by more than one codon, but a particular codon does not specify more than one amino acid. So the genetic code has redundancy but no ambiguity. For example, although codons GAA and GAG both specify glutamic acid redundancy, neither of them specifies any other amino acid. No ambiguity. The genetic code is universal. It is the same in almost all the organisms. For example, AGA specifies arginine in bacteria and humans and all other organisms whose genetic code has been studied. Because of the universality of codon, the genes can be transferred from one organism to another and be successfully transcribed and translated in their new host. The study of genetic code of mitochondrial DNA, however, showed that genetic code is not that universal. For example, UGA stop codon, AUA, AGA, nuclear DNA stop codon, isoleucine, arginine, mitochondrial DNA, tryptophan, methionine, stop codon. Thus, it appears that genetic code is not quite universal. Translation. Translation is the second stage of protein synthesis. Gene expression and translation messenger RNA messenger RNA produced by transcription is decoded by the ribosome to produce a specific amino acid chain. A polypeptide that will later fold into an active protein in bacteria. Translation occurs in the cell cytoplasm, where the large and small subunits of the ribosome are located and bind to the messenger RNA. In eukaryotes, translation occurs across the membrane of the endoplasmic reticulum. Although translation is a continuous process, but for convenience, we will discuss it in four phases: activation, initiation, elongation, and termination. After that, the product of translation in the form of amino acid chain polypeptide is formed. Activation of amino acids. Activation of amino acid refers to binding of free amino acid dispersed in cytoplasm to the three end of particular transfer RNA molecule. In this way, a complex is formed called aminosyl transparent RNA complex is formed. This binding is catalyzed by aminosyl transparent RNA synthase activation enzyme. Various amino acids that are to be take part in polypeptide formation have been continuously activated throughout the process of translation. Formation of maturation complex. Process of translation actually begins with the formation of initiation complex. It is formed by combination of ribosomal subunits, messenger RNA, a fast aminosyl transfer RNA complex, first transfer RNA molecules carrying a chemically modified methionine called N formyl methionine binds to the smaller ribosomal subunit. This binding is controlled by an enzyme called initiation factor. At the same time, 5' of messenger RNA molecule also bind to the ribosome. With the help of another initiation factor, initiation complex is completed when larger subunit of ribosome is also placed upon smaller subunit. Polypeptide elongation. In this phase, ribosomal units move along messenger RNA. Amino acids are brought by transfer RNAs, which are joined together to form polypeptide chain. This is accomplished by three steps, which are repeated again and again throughout this phase. Whichever codon of messenger RNA is exposed at A site, its anticodon bearing amino acid transfer RNA complex bind to it with the help of an enzyme, the elongation factor. Then an enzyme peptidyl transferase is emerged from P site. It removes the amino acid may be a chain from transfer RNA present on P side and bind it to the newly coming amino acid with the help of peptide bonds. Then the ribosomal subunits slightly move along messenger RNA from 5 to 3 direction so that a new codon is exposed at S site. This movement is called translocation. As a result, the empty transfer RNA is reached at E site to leave the ribosome while the other transfer RNA bearing a chain of amino acid is shifted from S site to P site and another codon is exposed to S site. These three steps are repeated again and again until the stop codon is reached at S site. Termination. Elongation continues in this fashion until a chain terminating nonsense codon is exposed at S site. Nonsense codon do not bind to any transfer RNA, but they recognize by release factor that terminate the process of translation and the polypeptide is released from tRNA. The tRNA is released from the ribosome and the two ribosomal subunits separate from the messenger RNA.